Hello everyone, this is Nath Goswami. Today we are going to discuss on the insect vision. So it is known to us that insects generally possess compound eyes for their vision. So this compound eye, this term itself is indicating that this is composed of or constructed from many similar units called Ommatidia. So the question is, do all the insects possess compound eyes? So they are present in most of the adult pterygota and the larvae of hemimetabolous insects. But they are strongly reduced or absent in parasitic groups such as Theraptera and Siphonaptera, and also in female coccids belonging to the order Hemiptera. Among Aterygota, the compound eyes are lacking in some Thysanura. Now, how many ommatidium can be present in a compound eye? So it can vary insect to insect. So there are up to 30,000 ommatidia or ommatidium in each eye of dragonflies, 10,000 in drone of honeybees, 5,500 in worker honeybees, and 800 in Drosophila. At the other extreme, the workers of ant, Ponera punctatissima, have only a single ommatidium on each side of the head. Now, we'll try to see the detailed structure of the ommatidium. So, although I am not a clay model artist, but I have tried to make one model of the ommatidium. So, if something is not correct in this model, I may be excused. So, you see this structure, and you can see that it is divided into two different major components. One is light gathering unit and another is light detecting unit. Now this corneal lens, this one is a transparent cuticle which is secreted by two modified epidermal cells called corneogen cells. After secretion of these cells, they differentiated into primary pigment cells, and below the corneal lens, this one is the crystalline cone. This one is the crystalline cone, the green one. This structure is secreted or formed by four cells called Samper's cell. Now the light enters the corneal lens and is focused on the light detecting apparatus by crystalline cone. The light detecting apparatus of each ommatidium contains 7 to 8 photoreceptor cells called retinular cells. These are actually photoreceptor neurons which can detect wavelengths of light. The light sensitive dendrite region of the retinular cells is rhabdomere. Together, all the individual rhabdomere are referred to as rhabdom. These all are together referred to as rhabdom. Now, one rhabdomere is consisting of parallel microvilli containing light detecting visual pigment molecules which are embedded into the plasma membrane. retinular cells are surrounded by 12, 12 to 18 secondary pigment cells, so that each ommatidium can be functionally isolated from its neighboring ommatidium. Now this ommatidium sits on a basement membrane in the retina, and the axons combine below the membrane to form the optic nerve that lead to the protocerebrum of the insect brain. Now we'll try to see how the insect compound eyes function during vision. So probably it is known to all of us that as far as insect vision is concerned, insects have been categorized into two broad categories. One, day active insects or diurnal insects, and two, night active insects or nocturnal insects. So the ommatidium, which is there over here, is belonging to the day active insects, and this ommatidium is belonging to the night active insects. So the day active insects have photopic or apposition ommatidium. In this case, the base of the crystalline cone is in direct contact to the rhabdom. So here the incoming light is focused on the base of the crystalline cone and directly onto the rhabdom. Also here the lights which are somehow focused on the angles are absorbed by the screening pigments located at the cytoplasm of pigment cells surrounded by this type of ommatidium. So the screening pigments prevent the light entering from one ommatidium to the adjacent ommatidium. Therefore, the light which enters in this type of ommatidium stays within that. And here, each ommatidium serves as individual visual unit. Now, another type, which is scotopic or superposition ommatidium. So this is almost similar to that of the apposition or photopic, except it has an open space over here known as clear zone, and it is there between the crystalline cone and the retinular cells. And this zone is actually containing the transparent crystalline tracts that arise from the retinular cells and extend to the crystalline cone. Unlike the photopic ommatidium, this scotopic ommatidium is sensitive to light intensity. So it is very interesting that during day it acts like photopic ommatidium, and during day the shielding pigments absorb the light rays which are there at angles and prevent those to enter into the adjacent ommatidium. But when the light is not abundant during the night, these shielding pigments or the shielding pigment cells are contracted upward, or they are distally contracted, and therefore the light rays which focus on the angles enter the adjacent ommatidium. They can enter the adjacent ommatidium and they create the multiple superimposed images on the rhabdom of the adjacent ommatidium. So this is the difference between the photopic or apposition ommatidium and the scotopic or superposition ommatidium. I hope that you have enjoyed the video, and I hope that the insect vision is clear to you. Thanks a lot. Thank you very much.